All right, well, good morning, everyone. It's a pleasure to be presenting you all today, to you all today as part of the ACES webinar series. So, go to the next slide. Seems very slow. Hang on, for some reason it's not switching. Okay, is that switched?
It has, yes.
Thank you. Right, so by way of introduction, I'm a senior researcher at UOW and a CI within the synthetic biosystems theme of ACES, where we're combining knowledge of cutting edge materials such as synthetic or natural biopolymers with our expertise in cell biology to develop a next generation of smart devices. What exactly does that mean? Well, we think of a smart device as utilising the advanced materials that we make and or apply in our labs to create new research and potentially medical devices. Now, on topic, my lab is particularly interested in neuroregeneration, which is required by the nervous system to either repair or replace nerve cells that have been damaged by injury or disease. While lower organisms have the capacity for neuroregeneration, evolutionarily higher organisms, including humans, are somewhat limited in their ability to regenerate nerve cells, which poses significant issues for treating both the central, that is the brain and spinal cord, as well as the peripheral nervous systems. We and others are attempting to address the problem by identifying ways to produce neural cells and 3D tissues from stem cells outside the body for possible application inside the body or for researching ways to drive within body nerve cell and tissue regeneration. Now, the latter involves tissue modelling for drug-based pharmaceuticals or electroceuticals, that is electric pharmaceuticals, discovery, disease and trauma modelling, and other methods of cell and tissue manipulation to then be applied clinically. Now, many of you are aware that stem cells offer significant opportunities to engineering tissues in vitro, as well as for targeting for in vivo tissue regeneration. Now, stem cells are defined by their ability to self-renew, as well as their ability to differentiate into multiple cell types. And so they can be used for replacement of damaged neurons. Now, traditionally, adult stem cells isolated from regenerative parts of the brain, such as the hippocampus and the subventricular zone, have served as a source of neural stem cells. Now, with the advancement of pluripotent stem cells, including embryonic and engineered human-induced pluripotent stem cells, or iPSCs, there's new and exciting approaches for neural cell replacement that are being developed. Now, in addition, with increased understanding of the human genome and epigenetics, scientists have been successful in the direct genetic reprogramming of somatic cells to a neuronal fate, bypassing the intermediary pluripotent stage. Whilst extensive hurdles still remain for the translational application of such stem cell reprogramming strategies, they're revolutionising the field of regenerative biology and providing innovative approaches to regeneration of the nervous system. Now, conventional methods of cell culture rely upon 2D planar culture, illustrated here on this slide by the first reports of research and clinical grade embryonic stem cells and iPSC culture, with the latter being awarded the Nobel Prize in Medicine to Japanese researcher Shinya Yamanaka. And notwithstanding their significance, the methods were reliant on planar or 2D culture of the stem cells as relatively flat colonies. More recently, the bringing together or synergism of stem cell biology and biomaterial technologies had a huge impact on stem cell based research and longer term clinical application for tissue engineering, including neural tissue engineering and in vivo regeneration. Now, biomaterials development is rapidly advancing with the discovery and use of biopolymers to display properties that in a really precise and physiological fashion can drive cell state and fate, both in vitro and in vivo. And so the design of novel materials is really trying to recapitulate the molecular events involved in the production, the interaction and the clearance of molecules within tissue, inclusive of the stem cell niche, such as the intercellular mass of a blastocyst for pluripotent stem cells, and again, the walls of the lateral ventricles and subgranular zone of the dentate gyrus in the hippocampus for neural stem cells, or the vascular niche in bone marrow for hematopoietic stem cells. Now, this applies to both pathologic tissue and regenerating healthy tissue or organs. And these events essentially translate to influencing cell behaviour and functionality by altering the morphology, adhesion, the motility, proliferation, endocytotic activity, protein abundance, and gene regulation. Now, at a most basic level, biopolymers can be used to recapitulate the role of extracellular matrix, or ECM, with the potential to influence spatially and temporally. The materials provide informative microenvironments mimicking a physiological niche, simply put, allowing the stem cells to interpret the polymer instructions and modify their behaviour accordingly. And so a biopolymer will transmit specific signals to cells that are decoded into biochemical signals. Hence, as for other biomaterials, the surface topography, the modulus or the softness, the porosity, and even electrical properties of, for example, a hydrogel or conducting polymer influences cell survival and fate, the latter acting via material actuation or intracellular ion, for example, calcium influx. Now, direct right printing of stem cells within polymeric materials presents a really good opportunity to engineer tissue for in vitro modelling and regenerative medicine. Now, here I've highlighted the first examples of constructing neural tissues by printing human neural or pluripotent stem cells that are differentiated within the 3D constructs to functional neurons and supporting neuroglia. In both cases, the constructs include a novel clinically relevant polysaccharide-based bioink, which we've named Triink, comprising alginate, carboxymethylchitosan, and agarose. The printed bioink rapidly gels by stable cross-linking with calcium chloride to form a porous 3D scaffold that encapsulates the stem cells for in situ expansion and differentiation. Alginate and agarose provide structural support for the constructs, with alginate enabling gelation in the presence of the cations after printing, and agarose conferring suitable bioink viscosity during printing prior to gelation. Now, the carboxymethylchitosan is a water-soluble derivative of chitosan, and it's conducive to cell survival within the construct. Differentiated neurons form synaptic contacts, they establish networks, they're spontaneously active, and they also show a drug-induced increased calcium ion response. For the iPSC-based construct shown on the right, in addition to neural cell induction, we're also able to demonstrate differentiation to what are known as embryoloid bodies, or EBs, comprising cells of the 3 germ lineages. That's endoderm, ectoderm, and mesoderm. And this, I can change it, provides proof of concept of developing all tissues of the human body, depicted here as part of an ontology tree for context in embryonic development. Now, in addition to bioprinting for 3D tissue fabrication, we're also utilising the natural self-organising capability of pluripotent stem cells on and within biopolymer substrates and matrices to form even more complex tissues known as organoids. An organoid is a 3-dimensional organ bud. It's grown in vitro and shows realistic microanatomy relative to the organ that you're trying to model. Cerebral or neural organoids are artificially grown miniature fetal brain-like structures that contain several types of neural cells, inclusive of neurons and supporting glia, and anatomical features relevant to early fetal brain. Now, the cerebral organoids are most similar to layers of neurons called the cortex in the brain and choroid plexus, although shown here in some cases they can include structures similar to the retina, meninges, and the hippocampus. In order to circumvent the shortfalls of conventional approaches or methods to generating cerebral or brain organoids, we've replaced standard mouse sarcoma, that is tumour-derived matrigel growth substrate, with more defined and clinically amenable gelatin methacrylate, commonly referred to as GelMA. GelMA is a versatile semi-synthetic matrix that incorporates the intrinsic bioactivity of natural matrices with the fidelity of synthetic biomaterials. It's an inexpensive and easy to handle derivative collagen that comprises both natural cell binding motifs, including RGD, the principal integrin binding domain present within ECM or extracellular matrix, and matrix metalloproteinase sensitive degradation sites or for proteolytic breakdown and cell migration, and various amino acid side chain functionalities for covalent modification. So additionally though, GelMA remains stable at 37 degrees, which is really important for tissue engineering. The differentiation of iPSCs is staged following initial culture on the GelMA, forming early neural precursors by day 5 after neural induction, neural rosettes and early stage neurons by day 14 following transition to neurodifferentiation media, and organoids or bona fide organoids with mature neurons by day 28. The organoids comprise densely packed cell bodies or cell soma that self-organise to hollow neural tube-like structures which represent the precursors to the brain ventricles, as well as neurons with prolific neurite outgrowth that form functional 3D networks with synaptic contacts. Now, consistent with recent reports of optimal substrate stiffness for iPSC survival and induction of neuronal cell fate and neurite protrusion, the GelMA is mechanically tuned for a softer gel that is less than 500 pascals, also resembling the physiological stiffness of brain tissue. Our optimised method provides a defined and much simpler platform for both research and translation of iPSCs, their neural derivatives and the organoids, including 3D in vitro modelling of brain development and disease and tissue engineering. Now, changing tack somewhat, you'll recall my earlier reference to the use of electrically conductive polymers for cell manipulation through electrical stimulation. While eStim has a long history in medicine, including pacemakers for the heart, cochlear implants for auditory function, and deep brain stimulation for disorders like Parkinson's disease, it's only really recently been considered for modulating human cell behaviour in vitro for tissue engineering and modelling and regenerative tissue in medicine. Now, conductive polymers such as polypyrrole are amenable to in vitro and in vivo cell stimulation, being easy to synthesise with different counterions, that is dopants, to augment biocompatibility and cell effects. And more broadly, the utility of eStim isn't particularly surprising, given that our bodies are naturally bioelectric, with organs and component tissues operating electrically for normal physiological function, including during development and healing following injury. So, several years ago, we demonstrated that polypyrrole could be used to electrically stimulate human neural stem cells and augment their differentiation to neurons when containing the anionic dodecylbenzene sulfonate, or DBS. Now, for those not familiar with polypyrrole, it's a type of organic polymer formed by the polymerization of pyrrole, which is with its oxidized derivatives being good electrical conductors. Now, interestingly, stimulated cell cultures comprise higher and lower induction of neurons and glia respectively, with the neurons forming nodes or clusters, as well as having longer neurites with greater branching compared to unstimulated cultures. Now, importantly, cell clusters showed a similar spatial distribution to regions of higher conductivity on the polymer film surface, which suggested the level of conductivity and therefore stimulation influences cell adhesion and/or galvanotaxis. While for now we can only surmise, our findings are consistent with using modulation of conductivity and electroactivity of polypyrrole or other conductive polymer-based substrates used to grow and stimulate cells to regulate the maturation and organisation of neural networks in vitro and in vivo. Now, extending our earlier film-based 2D or planar studies, we recently reported using electroactive polypyrrole with DBS to modulate fate determination of human iPSCs. Again, it wasn't particularly surprising to find that eSTIM similarly augmented neural induction of iPSCs when combined with the usual cocktail of chemical inducers. However, remarkably, stimulation without chemical inducers, that's neural inducers, resulted in iPSCs differentiating to cells of the previously mentioned three germ lineages, endoderm, ectoderm, and mesoderm. A phenotypic characterization further showed robust induction to neuronal fate with electrical stimulation, again without the customary chemical inducers. Our findings add to the growing body of evidence for using eSTIM to independently augment stem cell differentiation, more specifically pluripotent stem cell differentiation and especially neuronal induction. In addition, we've shown the versatility of electroactive polypyrrole as a cell-compatible platform for stem cell research and translation, including identifying novel mechanisms of fate regulation and tissue development relevant to electroceuticals and, again, regenerative medicine. An investigation of the fundamental molecular mechanisms that underlie the effects of stimulation governing differentiation will be really useful. For example, brain-derived neurotrophic factor, otherwise known as BDNF, being a member of the neurotrophin family of growth factors and important for neuronal maturation, growth, and survival. It's been shown by us, again, using the polypyrrole film platform and others, to be responsive to electrical stimulation. So correspondingly, electrical stimulation via the film elevates cell surface expression of BDNF receptors, specifically its high affinity tyrosine receptor kinase B or TREC-B, and enhances the trophic effects of BDNF on neurons. Although the involvement of BDNF in stimulating human iPSCs remains to be determined, animal cell modelling further suggests increased BDNF expression by first initiating an action potential via polymer-mediated stimulation, which in turn triggers calcium ion influx across the cell membrane and subsequent activation of protein kinases, phosphorylation of cellular transcription factors, and finally transcription of the BDNF gene. Interestingly, the recruitment of intracellular TREC-Bs to the plasma membrane of cells is believed to be gated by key intracellular signal transducer cyclic AMP following stimulation. Now, if you're interested in further detail about current understanding of the cellular mechanisms underlying eSTIM, just to put it out there, we've recently wrapped up a chapter titled Electroceuticals for Neuroregenerative Nanomedicine. It's for a book that's to be published by Elsevier, which includes an up-to-date overview. So anyone that's interested, feel free to email me if you'd like further information on that. But mechanisms aside, following on from the broader topic of using conductive polymer films for 2D cell culture and stimulation, so as to better model 3D native neural tissue development, regeneration and function, in collaboration with Professor Jadranka Travisevich and now Dr. Pico Zhang. Pico recently, I believe, graduated from his PhD studies. They're from the University of Auckland. We've more recently progressed to interfacing 3D printed conducting polymer pillar electrodes prepared as arrays from polyethylene dioxide thiophene, or PDOT. It's a bit of a mouthful really, and a dopant polystyrene sulfonate, or PSS, with human neural stem cells encapsulated within the previously mentioned trianc biogel. That's the gel that comprises the alginate, agarose and carboxymethylchitosan. Now first, the electrochemical properties of the pillar electrodes and the biogel support their electrical performance, with the latter shown to have well-defined electrochemical responses through ionic conductance with or without cell loading. Now, second, consistent with conventional plate-based culture, gel-laden cells differentiated on the pillar arrays into neurons and glia with or without stimulation. However, while unstimulated constructs were relatively poorly maintained at the interface of the ventral surface of the constructs and the insulating substrate on the array surface, stimulated constructs developed into tissues with large numbers of really densely packed cells abutting the underlying substrate, with polarized neuronal cells exhibiting axons and dendritic arborizations. So if you look at that panel of images on the bottom of that slide, the image on the left is unstimulated, and the image in the middle is the stimulated cells. Now, even more interesting, deeper tissue analysis revealed tracks of intensely labelled and high-density mature neurons with synaptic junctions, shown by the arrows in the bottom right image. And it's suggestive of structured neuronal network formation and tissue maturation. A quantitative analysis supported qualitative assessments with indices of neuronal maturation indicating more mature neuronal tissue for stimulated compared to unstimulated samples. And finally, cellular networking and function of both unstimulated and stimulated constructs was supported by recurrent spontaneous increases in extracellular calcium concentration. Also, disinhibition of cells by the inhibitory neurotransmitter GABA receptor A antagonist by calculin resulted in an increased firing rate and spike amplitude of stimulated tissues compared to unstimulated constructs, which is consistent with enhanced maturation of neural networks and a possible increased responsivity to pharmaceuticals, which we're now investigating further. So to summarise, the printed polymer pillar array platform is really promising for bioengineering both 3D neural and likely non-neural tissues by enhancing maturation and function. In addition to tissue engineering, the platform may inform within body stimulation for regenerative medicine application. And finally, it's worth noting that this work has helped advance our electric tissue concept, for which we were awarded a Research Australia Frontiers Research Award last year. So clearly polymers, either synthetic or natural in origin, are being extensively evaluated for cell support, with our own work indicative of human neural cell support and related tissue engineering. They offer a wide range of versatility, in particular regarding shape and mechanical characteristics. I think it's safe to say that their biocompatibility is really unmatched by other biomaterials, such as metals and ceramics. And several recent studies have shown that polymers can be shaped into suitable support structures, including nerve conduits, scaffolds, and even electrospun matrices capable of improving the regeneration of damaged neural tissues. Excuse me. So while currently most neural tissue applications are in preclinical study, there are examples of collagen polymer conjugates aimed at regeneration of peripheral nerves being successfully tested in clinical trials. In closing, polymeric applications for neural tissue will no doubt continue to evolve, with our own focus in particular on biocompatible electro-conductive materials such as polypyrrole, and the use of growth factors and stem cells combined with polymeric structures will no doubt or inevitably improve the restorative capacities of the materials, as well as their ability to target specific areas of the nervous system, inclusive of being used to deliver therapeutic agents. So right now, we've only really the tip of the iceberg with the opportunity to discover innovative combinations of biopolymers, cells, and other agents such as proteins towards new real-world therapeutics and regenerative applications. So thanks to you all for your time today, and I'm happy to take any questions. Thank you.
Thanks so much, Jeremy. Gordon has popped a question in the chat there. Would you like me to read it out, or are you able to see it?
Got it. Thanks for a great talk. Thanks, Gordon. Any comments on approaches to optimize the eStim parameters, e.g. when to apply? Yeah, really good question. I guess, Gordon, that is where we need to go next. We've been using a well-worn protocol to date to stimulate our cells and tissues in vitro, and whilst it works and shows an effect, I would imagine it could be further fine-tuned. It may, whilst we've been applying it for primarily neural tissue induction from the various stem cell types that we're working with, in terms of developing other tissues using eStim to augment, it may be that we need to modify the parameters that we're using to get the result that we're after. And of course, when we jump from the bench to the bedside and start trying to apply some of these stimulation platforms in vivo, it may be, again, quite different in terms of the level of stimulation, the timing, as you say, frequency, pulse magnitude, etc. So I guess there's a lot of scope for further development here, and for me, I think that's really exciting. A lot of opportunity.
Yes, thanks.
Any other questions?
Yeah, I had a question, Jeremy.
Okay.
So with the 3D pillar printed electrodes, you were saying that the neural tissue was differentiating along the electrodes, kind of aligned with the electrodes. I'm just wondering if you think that there was an effect of either current density or the topography of the electrodes themselves that might have caused that
another
response.
good question. I would say both. We do have plans to look at variations of the electrodes in terms of the shapes of the electrodes. The current platform, as you mentioned, utilizes pillared electrodes. One of the questions that we got from reviewers when we were finalizing the paper that we published relates to or related to varying the shape of the electrodes, pyramids, and all sorts of different conformations, and to investigate the effects. We really like to do that, and we no doubt will, in collaboration with Jadranka, who has the expertise to be able to vary the electrodes in that way. So I think I've answered your question, but again, you know, there are a lot of unknowns there, and you know, again, it's really the tip of the iceberg.
Yeah. Okay. Thank you.
Mm-hmm. Okay, well, I guess if there's no other questions, please don't hesitate to contact me after this, email or otherwise, if anybody has anything they'd like to ask about you know, the work that we've done and we're doing. And you know, again, thanks very much for coming online and listening to me.
Thank you.
Cheers.
Thanks
Thank
so
you,
much,
Jeremy.
Jeremy.